Thank you for joining us today as we explore a fascinating and rapidly evolving field that merges the principles of pharmacology and genomics. I am Eshika and I will be moderating today's session. Our topic for today is pharmacogenomics, a key component in personalized therapy. So, what exactly does the term pharmacogenomics mean? Well, it is the study of how an individual's genetic makeup influences their responses to drugs. This field combines pharmacology and genomics to develop effective, personalized medications with minimal adverse effects. By understanding the genetic variations, pharmacogenomics aims to predict which medication is correct for which disease. This personalized approach, therefore, can improve the treatment outcomes for each patient immensely and find the right medication for the disease condition. Today, we are honored to have with us Dr. Risha Khandelwal. Dr. Risha is a scientist at DrOmics Labs, Mumbai. She is a distinguished researcher in the field of precision medicine with over 9 years of experience. She holds a PhD in basic life sciences and has furthered her expertise in applied cancer research with a postdoctorate from one of India's premier cancer research institutes. She specializes in cancer research with a focus on prognostics and therapeutic solutions. Her expertise lies in personalized genomics, where she has made significant contributions to understanding how an individual's genetic profile can be used to find tailored treatment strategies. Welcome, Dr. Risha.
Thank you so much, Ashika, for the nice introduction of myself and the pharmacogenomics both. I think my initial part you only explained, so I think the audience is pretty warmed up for the pharmacogenomics. Thank you.
All right, ma'am, my pleasure. So, Dr. Risha, I will start away with the questions. My first question to you is, can you explain the basic principles of pharmacogenomics and how it enhances our ability to predict drug responses?
Okay, thank you. Thank you for starting with this question, because since in my reviews in last couple of years, I found that we need to create awareness about the basic terminologies of pharmacogenomics first, and then people need to be discussed about the pharmacogenomics. So when we talk about pharmacogenomics, as you have already explained, it is a combination of the pharmacological principles and the genetic information of a person. Basically, what we are doing is we are trying to see how these two information are interacting with each other so that we can provide a very tailored, customized treatment to a person to reduce any adverse side effect, to give a very customized, tailored treatment and dose adjustment. So when we talk about principles, I will take you through the basic terminologies which warm us up for understanding the further information. So very first thing, as you said, Ashika, it was a genetic variation. Genetic variations are the DNA changes in terms of single base pair, insertions, deletions, right, and duplications, which will bring about changes in some of the very important pharmacological genes, the genes which are actually processing the drugs and making the drugs available to the body and excretion of the body, and more so over. So genetic variations are studied primarily in pharmacological important genes. The discovery and the innovation is still going on. Second, a very important aspect, the pharmacological aspect, I wanted to explain that when we talk pharmacological as principles, the major factor which decides a person's drug response will be how that person is processing the drug, which comes under pharmacokinetic. Kinetic, as the name suggests, that kinetics of the drug through your body. So it has 4 fundamental principles. Okay, so how the drug is absorbed, distributed in your body, the metabolism of the drug, and how the drug is excreted. So I'll go through one by one. So when you talk about absorption, it's primarily when we consume a drug. Okay, so how it is taken inside the blood flow of the body, it is the process of absorption. It is a process which is primarily regulated by various drug transporters and enzyme proteins. So if I want to give an example where absorption can hinder a particular drug response, is a very nice example I would like to give simvastatin. We all know that cardiac diseases are the next epidemic in the country. Even Lancet has published it. Okay, statins are the very important medications which are used to control the cholesterol in that. So there is one gene called SLCO1B1, which hinders the absorption of the drug inside the liver. And that is why what happens is it accumulates in the blood and causes a toxicity. So when you talk about distribution is once the drug is in the blood, it has to go to its actual site of working, right? So so one example is, so we know that the one more major concern in our country or globally is, depressive disorders or neurological disorders. So many of the major depressive disorder drugs are not able to show effect in more than 50% of the people. And I'm not telling from what I feel. I have studied the research papers, and one of the very important drug which can influence the passage of the drug to the brain is a MDR1 gene. Basically, it doesn't allow the drug to cross the blood-brain barrier. So we can say, okay, simply, people will sit in the corner and they will sulk for more time if they are not, the medicine is not working. But no, studies have found when the efficacy is low for these drugs, during that time, the tendency of the suicides have increased. I mean, I still shiver while talking this sentence, but that is the truth. People have done the study on that. So this is the distribution. Thirdly is the metabolism. Yes, the drug has to be metabolized, converted to a simpler form so that it can be excreted by the kidney. And the major site for the metabolism is the liver. And the liver enzymes play a very important role in converting to simple metabolites. And mutation in these enzymes can divide the person who are poor metabolizers of a drug. What will happen when they're poor metabolizers? The drug will not get metabolized. It will keep accumulating in the body. And what will happen? It will cause a toxicity, right? And opposite is the case for the ultra rapid or rapid metabolizer. So what will happen? People will, take the drug. It will be tremendously metabolized, very fast, in a very fast manner, and it will be excreted. So what will happen? The desired effect or efficacy which they were expecting will not be coming. And now I'll move. Sorry.
No, please continue. I was just agreeing with you.
Yeah. So and now I will move to the final part of the ADME process, which is excretion. Excretion is excreting the blood, the drug metabolites to the urine. So even there are some renal transporters like SLC22A2, which we all know a very common magic drug called metformin, which is used to manage diabetes in globally or in our country. And many of the people, because of some mutations, actually they excrete metformin so fast that they're not able to get the desired effect. Many of the people, they say, oh, I'm taking metformin, but I'm not able to see the effect. The major reason can be that metformin is not even having what we say enough turnaround time. It's just simply getting excreted. So there are certain mutations in Caucasian and African American population where in the this gene, because of which metformin is excreted very quickly. So yes, so I try to simply explain the principle of the pharmacogenomics and its effect in terms of what we say, how these principles can interact with the drug effect. Now, coming to the second part of your question, Ashika. So how pharmacogenomic enhance the drug response prediction? So as I just said, before go ahead, I try to gather, or I have put forward information about the major concerns of our country. There can be hundreds of examples I'm saying here, but I'm focusing on the cardiology, depressive disorders, and cancer over here, because they are the haunting devils in our country primarily. So first is, so how pharmacogenomics can help us is, very first thing is a very personalized medicine, right? So when we talk about cancer, every cancer they have different what we say, molecular driver. For example, if you take breast cancer, not all breast cancer patients are same. Some of them can have different mutations, like for example a HER2 or ERBB2 positive breast cancer. In that, if you find HER2 what we say mutation, trastuzumab medicine is recommended for the patient. It's a very specialized medicine. Second most important is dosage optimization. So we all know that cancer patients suffer tremendously while taking the chemotherapy. And many of the patients, we don't know why only some patients are having toxic effect and some patients are not having toxic effect. So I will give you just one example. There is a gene called TPMT. If the activity of the gene has gone down, because of which the thiopurine drugs which are used for chemotherapy they are metabolized less. The patients become poor metabolizers of the drug, and because of which the drug accumulates, and because of which patient have to face the toxic effects of the this thiopurine drugs. So we have gone through personalized medicine and dosage optimization. Thirdly, I would like to take you through obviously reducing trial and error. So this is especially for the conditions where you do not have much scope to do the trial and error. And in the last but not the least, improving the drug safety. So when we talk about drug safety, we simply think, okay, some of the drugs might have some, chota mota side effects and all. For example, I have two examples actually to discuss over here. Is First example is I would like to mention atorvastatin. Atorvastatin is a drug which is used to manage the cholesterol in the patients. And what happens is many of the patients, so basically 6.5 crore prescriptions are given in a year in India actually for atorvastatin. And many of the patient experience muscle pain or myopathy after taking that medication. Okay, so there are certain genes which are actually causing this side effect, because of which atorvastatin is not suitable for the patient. So they are actually, after genomic testing, with some of the clinical, some of the genomic testing have recommended alternative drug for the atorvastatin, right? So basically, and secondly, we do have CYP2D6. CYP2D6 is one gene which is responsible for metabolizing or interacting with 25% of the major important drugs. And many of the times, because of certain duplications or copy number variations in CYP2D6, you need to prescribe alternative drug. So thus, we need to improve drug safety using pharmacogenomics. And lot of patients can be spared from the adverse side effects, or they can be spared from the trial and errors, or they can be given a proper dosage of their medicines. Yeah.
Okay, ma'am, that was an immensely detailed explanation, and I was really fascinated to know that there are so many intricacies still, and there is the gap amongst the common people and the research group in order, like, for the different kinds of drugs and etc. and the conditions. Okay, so my next question to you is, what types of genetic variations are commonly analyzed in pharmacogenomic testing?
Yes. As I have spoken to previously, that in pharmacogenomics, primarily genomic variations are studied which affect the drug metabolism or drug processing by our body. They can be present in coding or non-coding region of the DNA, first of all, and majorly they are present in the coding region. So in the coding region, and secondly, the kind of genetic variations are, they can be single nucleotide polymorphism. Now what this single nucleotide change can cause, they can increase the function of a protein, they can decrease because they can change certain DNA binding domain or drug interacting domain, or they can, what we say, completely truncate a protein. They can just bring simply the stop codon, because of which protein is not expressed at all, right? And this teeny tiny SNPs bring a lot of challenge in like drug processing in our body, but they are very important also. So if I have to give you one example, there are certain SNPs, okay, in the gene called CYP2C19, and this gene is responsible for processing of a very important drug called clopidogrel. Clopidogrel is an anticoagulant. Okay, it is used in managing cardiovascular diseases, and it is the essential medicine given by WHO as well as Indian government. It is available, many of the India's primary, tertiary, secondary care centers. Okay, now the fascinating part is, almost up to 30% of global population, okay, can be poor metabolizer of this clopidogrel, and 18 to 20% of the Indian population also can be a poor metabolizer for the CYP2C19, which is a gene metabolizing the clopidogrel. And that is why patients have to face the very serious adverse side effects. I mean, for like information, when clopidogrel causes side effects, patients are, told to use alternative medicine called ticagrelor. So apart from that, so that was about alternative medicine. These SNPs can lead us to find a very right dosage for certain very important medication. Now I'm talking about the second medication, which is primarily used in the country or globally for managing cardiovascular diseases, but it has very serious side effects and it has a very narrow therapeutic index. For example, what is therapeutic index? When you take a drug, it has a very specific concentration at which it will be useful for the person, for a particular medical purpose. So the drug concentration lesser than that, if it happens, then the drug is not effective, and drug concentration happens more than that, the drug is toxic. So certain drugs like antibiotics and all, they have broad therapeutic range, but certain drugs like warfarin, if a patient gets slightly more concentration than the patient requires, he starts internal bleeding, because warfarin, they are exposed to the higher concentration of warfarin than they require or than they can process, and there is over blood thinning and patients start bleeding, internal bleeding. So for startling statistics, 10 to 20% of global population and 10 to 15% of Indian population have this warfarin interacting genetic mutations and SNPs, and that can tell the gravity of scanning these SNPs in globally as well as at our country before administering these drugs. So after SNPs, I would like to talk about the second category of the mutations called copy number variations. So copy number variations, as it says, that during replication, certain genes are generate multiple copies, or they can reduce the copies or they can delete it. So in that case, what happens is level of expression of the gene changes. So CYP2D6 in one is a one gene which, as I already told, it interacts with numerous medications like antidepressants, pain relievers, right? So for our surprise, in India, 1 to 2% of the population may have a CYP2D6 duplication, leading to ultra rapid metabolizer. What is ultra rapid metabolizer? I will just repeat again. A condition where patient is over metabolizing a particular drug, because of which you can't see the effect, because drug is not staying in the body for the sufficient amount of time. So as I said, globally 1 to 2 like in India, 1 to 2% population can have this duplication, and globally 5 to 10% people may have deletions, leading to poor metabolizer. Okay, so that tells the gravity of including the duplication strategy in the genomic profiling, copy number variation detection in our genomic profiling. And the third category, as I told, insertions and deletions. Now, this insertions and deletion, as its name indicates, like certain fragments of the DNA are inserted or deleted during the process of the replication of the DNA. And yes, there is a gene which is as again I am telling about the cancer therapy. So UGT1A1 is a gene in the promoter site of that gene. It's not a non-coding. So this is one of the rare example where a pharmacogenomic mutation is found in a non-coding region. So promoter of this gene has some insertion, due to which a person has to take alternative drug for the chemotherapy. It has been proven in the studies. So thus we have different types of genomic variations, and types of genomic variation, they have to be screened very efficiently during our genomic sequencing to know accurately about their presence in the patient's DNA, and they play a very important role. Yeah, thank you.
Okay, ma'am. So my third question to you is, despite the promise of pharmacogenomics, what diagnostic barriers need to be overcome for more widespread adoption?
I would say I would like to start upon being a scientist. I would like to start first scientific what we say challenges which we have to face. So as just we mentioned, we spoke about, Ishika, that kind of different DNA variations we need to screen for the pharmacogenomic what we say profiling of a person. Many of us know that ethnicity wise or country wise there can be what we say multiple mutations which are present. So right now, majority of the studies for the pharmacogenomics have happened in Caucasian or European population. Yes, they do have a set of mutation which there are publication which 60-70% overlap with our set of mutations, but there are ethnicity specific additional mutation as well. So while implementing the pharmacogenomic testing, okay, so we do have to include what all mutations are studied and well defined for their pharmacogenomic effect, but we need to also investigate additional mutations which can be very much specific in our country. For example, as I was telling you about the metformin, SLC22A1 gene. Okay, so there are mutations in those genes which have very different frequency from the global population. Like for example, there can be some mutations which are very important in one country, but they can be less important in our country, and there can be some mutations which are very much important for our country. So why I am talking of this? Because many of the pharmacogenomic tests are performed on microarray, majority of the companies, because it can be cost effective and accurate way, and it is a time saving also. So while designing a test, while choosing the collection of the mutations, we need to be very careful that we are including the globally accepted mutations, plus we are also trying to simultaneously find what can be the novel mutations which are specific for the Indian population. We have to keep our eyes open for that. And apart from that, there are technological barriers. Like as I've told you, that pharmacogenomic testing is done either on a microarray, where you have certain specific set of the mutations, or it can be done in whole genome and whole exome sequencing. The most acceptable practice is whole exome sequencing, where you're sequencing the entire coding part. But the fact is, in the industry, many of the companies are using the microarray, where particular sets of mutations are there, and we need to include comprehensive allele selection or mutation selection in that. Now let's come to on ground. The problem on the ground actually is way more a bigger devil than the scientific problem I just told about, because we do have a very decent data about the pharmacogenomics, but the very first barrier, I would say, in a developing country like India is the cost. So this cost can really deprive rural and lower economic group from accessing the pharmacogenomic test. Secondly, I'm talking about a system which needs a more sophistication. For example, we are still not maintaining that much electronic health records. So basically, when people are tested for like genomic testing, the testing results had to be integrated in a sophisticated electronic health record, so that the clinicians can access it. For example, you put an ID and you access the genomic data, and so that patients can get real-time consultation. For example, in Japan, they have set up a Biobank Japan project. So what it does is it collects genetic and clinical data for research purposes and integrates it into the EHR, so that it becomes easy for people to get a real-time data, as well as R&D ke liye bhi it helps a lot. But in India, what happens is, in our country, I mean, which we can, what we say, really, I'm looking forward to see is, we should have a robust IT system for protecting genomic data and integrating the clinical data into electronic health records. And we need to train people to do all those stuff. So also one more aspect is awareness in the clinical practitioners. So there is very interesting survey which was actually conducted in North Indian doctors. Out of 400 doctors, 35% of the doctors actually were knowledgeable about clinical applications of the pharmacogenomics, and surprisingly, only 13% showed interest in learning more live about applying the pharmacogenomics. So this was on ground, what we say, kind of barrier. One more barrier actually I recently came across is, there is one interesting, what we say, study approved in 2015 by Indian government. That study is clinical pharmacology. So basically, this is a very special stream of pharma studies they can be a very good mediators between the doctors and the patients. They can be actually knowing how the different drugs are interacting, and they can advise doctors. Because see, one patient goes to a doctor, he might be going to several different doctors, and so different medications that person is taking. So whatever interaction can be happening due to the different medication as well, it has to be assessed. So I think we need to seriously think about pulling in the pharma students, with us, to become a very strong bridge between the pharma industry, medical practitioners, and the patients.
Yeah.
Okay, because right now there is a huge knowledge gap, and of course everybody will have their own apprehension, but if we have a very qualified person bridging all the gaps, it will be very good. And apart from that, is if you see the actual implementation at a very large population set, you have to face regulatory approval problems. I mean, this is a genetic data. You need to do genetic testing. You have to go through the very complex approval system. And if you see globally, country wise, these approval guidelines can be different. Okay. And secondly, as far as India is concerned, the regulatory scheme, what we say, carving, is still in a very nascent stage. So, and regulatory and ethical barriers may, like, secondly, ethical concerns. So globally there are ethical boundaries drawn, like for example patient consent, and then genetic data sharing and all. But those, even the ethical boundaries or ethical guidelines are also different country wise. In India, uh, live about, like, what we say, in our country we need to be more aggressive about carving this ethical guidelines, so that pharmacogenomic implementation can take a speed. Yeah, so these are the actually problems which I could, or you can say barriers which I could think of.
All right, ma'am. So,
Yeah.
with this, we come to an end of today's session.
Hmm.
Um, it was great having you and your insights, especially on the genetic variations and the single nucleotide
Hmm.
polymorphisms and the copy numbers, and how these specific mutations, are affecting the, working or administration of different kinds of drugs, which, especially for the cardiovascular diseases, which are the need of the hour. So it was really insightful, and it was really interesting to know that the field of pharmacogenomics is so much required to advance in a country, in a developing country like India. And I'm sure that with your perspective, our audience will be, more than delighted to know about and gain a broader overview of the strong potential that the field of pharmacogenomics holds in precision medicine. So, with this, I would like to conclude and again extend my immense gratitude to you for sharing your thoughts.